the scientist Miller, it's called Miller and Urey experiment, and they, in their lab, they designed an apparatus that simulated, that means that copied the conditions that were present on Earth at that time, and that they kept that apparatus running for a week, okay, and then finally they wanted to see if any organic, molecules had formed. So what they did, this is their apparatus. They had water here in a round bottom flask. They're heating it because the earth was still very hot, so the ocean water was bubbling, okay, temperature high, so water vapor being created. And what was there in the atmosphere in earth at that time? This is NH3 means ammonia, CH4 means methane, and H2 is hydrogen gas, okay. So they artificially supplied these gases into the chamber, and since there were a lot of lightning strikes at that time, they gave electric sparks which were simulating lightning. So you see here, this atmosphere here is, the conditions here are very similar to what was present in on Earth at that time. Water vapor, hydrogen, ammonia, lightning strikes, and so on. And then whatever chemical reaction, then they kept it running for a week. Whatever chemical reaction was happening, the product that was coming out from there, they started collecting it, and they passed it through a condenser. Condenser means a cooling mechanism. They cooled it, okay, by passing cold water. They collected it. Whatever was, they didn't know what is being made here, okay. Whatever is happening here, they are just simply whatever gas is being created. They condensed it, cooled it down. They collected it in a conical flask, and then they analyzed it. So let's see if in this liquid that we are collecting, what are we getting? Are we getting any organic monomers? Are we getting any organic molecules? And to their surprise, they found amino acids and nucleotides. So these conditions, this was proved that conditions like this could lead to the formation of organic monomers, that means organic small molecules, from totally inorganic materials, right? So this was the first step. Now there are other hypotheses also, like life could have begun in volcanoes which were submerged, which were under the sea, or meteorites came from outside, from the space, they brought some material. Those hypotheses also exist. Now now they have shown that the conditions were such that monomers could have formed. Now how did the monomers polymerize? How did they join each other? Now researchers took these monomers and they started dripping, drop by drop. They started pouring it onto hot sand, clay or rock, because the earth was at that time very hot. So it was hot sand or hot, clay, whatever is there. And if you pour this liquid, the liquid that was connected here, which contains monomers, you take this liquid and you take a dropper and you start dropping the drop by drop. You put this liquid onto the rocks and you start collecting what is forming. They found that the monomers polymerized because the high temperatures helped in the polymerization reaction. That means the first simplest protein, the first simplest RNA was made. And actually it is believed that before DNA, it was RNA that came. RNA are short, RNA are unstable, but that was how the first genetic material was actually not DNA, but RNA, very short strand of RNA. So stages, you see here, we were talking about 4 stages. Synthesis of monomers, small organic molecules, possible, already shown by research. Joining of the small molecules to form big macromolecules, possible, they showed it by research. Now the third is packaging these molecules into pre-cells. That means cell membrane has to form. So the researchers have already demonstrated that when you have these, what is the cell membrane made up of? Phospholipids, fatty acids, okay? So now that polymers had formed, so lipid polymers that had formed, if it could somehow come into contact and because of surface tension and other changes, somehow it, by chance, it surrounded a protein or an RNA material inside and became a sphere. That was the starting of the cell. That's a pre-cell. Okay, so they have demonstrated that pre-cells could have formed spontaneously from fatty acids. if you remember, this is the glycerol molecule. This is what a triglyceride fatty looks like. You see the fatty acid chain. So the fatty acid chain is what was responsible, and this formed the first pre-cell. Now the last stage is self-replication, means cell division. That means making baby cells. So mechanism of information flow probably emerged slowly, baby steps, okay, through a series of small changes to much simpler processes. And that is why scientists, the hypothesis is that the first genes were actually not DNA genes. Those were RNA genes, short strands of RNA. Because if you remember, when I taught you DNA replication, RNA transcription, there were a lot of enzymes involved, right? Anybody remembers the name of any enzyme that helps in DNA replication? Yes?
DNA ligase.
DNA ligase. What was the most important one?
DNA polymerase.
DNA polymerase. So what are those enzymes? They are proteins. So if you're thinking of genes and replication, DNA replication, that means if DNA replication had to happen, we needed those sophisticated enzymes. We didn't have the sophisticated enzymes at that time. So the researchers believed, no, it was not DNA, it was RNA. And because RNA replication requires simpler things, it doesn't require as much of the protein enzymes, and actually simplest form of RNA replication can take place by RNA based enzymes, okay? So that is what might have happened. DNA was not there, it was RNA, very short strands, very unstable, and they were replicating without the help of proteins. Perhaps they used RNAs that can act as enzymes like ribozymes. So this is what was the beginning of life. Monomers formed, monomers joined to form polymers. By chance, a polymer got surrounded by a fatty acid chain, okay, that formed the first membrane, that was the first pre-cell. And the polymer that was inside the pre-cell is a nucleotide that made the RNA. So that was the first pre-cell. We don't even call it a cell. We're calling it a pre-cell, like a precursor of a cell. And that RNA started replicating. Now, as that pre-cell started replicating, that we started making baby pre-cells, natural selection, always present, right? Natural selection started acting on the pre-cell, started shaping the properties of those self-replicating RNA molecules. At some point during the millions of years of evolution, DNA, which was a more stable form, it replaced RNA. So DNA evolved from RNA, from a single-stranded RNA to a double-stranded DNA is an evolutionary modification, right? So they replaced RNA as the storehouse of genetic information, and then the cells became true cells, from pre-cells to true cells. So this is the hypothesis for the first genes. These were RNA monomers, right? Remember when you remember the apparatus? This is how the RNA monomers were formed from inorganic stuff, from methane, from ammonia. Then the monomers joined to form polymers, okay, big ones. These are polymer simple genes. This is assembly of a complementary RNA chain. Complementary RNA chain serves as template of the original gene, and this is how DNA evolved. So prokaryotes are everywhere. If you take the collective biomass, that is, like, if you weigh how much prokaryote is there in the world, it is 10 times that of all eukaryotes. So you can take all the whales, all the elephants and everything in the world and weigh them together. Their total weight is 10 times less than the total weight of all the microbes, all the bacteria, all the unicellular organisms that we have. Our microbiota, it helps us extract nutrition. That is one of the most important things that they do. They help us to survive. Without our microbiota, we might die. Prokaryotes also do us a big favor by decomposing dead organisms. If it was not for the prokaryotes, the earth would be full of dead bodies, no decomposition, no space for living animals. So they help us in decomposing, and they return the minerals in the simplest form back to nature. Okay, so they help in recycling. So prokaryotes are very essential. This is just a recap if you have forgotten what a prokaryotic cell looks like. This is a comparison of prokaryote versus eukaryote. Remember, very important. No membrane bound organelles. Membrane bound organelles, they do not have the modern nucleus. They have modern nucleus because theirs is called a nucleoid region, which is a primitive kind of nucleus. Now, classification of prokaryotes, broadly, we classify them into these three types based on their shape. So, if they are spherical, if they are round, we call them cocci. If they are rod-shaped, cylindrical, we call them bacilli or bacillus. And then if they are curved, comma-shaped, then we call them spiral. These are the three main types. Now, although all prokaryotes are unicellular, sometimes the prokaryotes live in colony. So when they live in a colony-like fashion, they tend to behave as multicellular organisms. We are multicellular organisms. What is our advantage? Our advantage is that because we have multiple cells, we have thousands and millions of cells, we have a division of labor inside our body. The digestive system is doing the digestion, the respiratory system is doing the respiratory work, the circulation system, cardiovascular system is circulating the nutrients and all of that. So we have division of labor. For a unicellular organism like bacteria, the single cell has to do all of their life supporting activities by itself. So during evolution, the prokaryotes realized that if they survived together, close to each other, like clinging to each other as a colony, there could be some kind of division of labor. The bacteria or the prokaryotes on the outside of the colony could be taking care of the defense. Okay? Then the bacteria or the prokaryotes on the inside, they didn't have to worry about protecting themselves. They could focus on the nutrition, right? So there was division of labor. Now, about half of the prokaryotic species are mobile. How do they move? There is this particular tail-like structure called flagella. Remember that only one kind of human cell that has a flagella? The sperm. So unicellular, in our body also there is one kind of cell. So a lot of the prokaryotes have flagella. In many natural environments, the prokaryotes, what they would do is they would attach to surfaces in a very organized colony called, what they do is called a biofilm, okay, which may consist of several species of prokaryotes. In our teeth, that is what happens. So each time we are eating food, there are bacteria present in our teeth. They exist as biofilm. They like to feed on the sugars that we've eaten. So they take those sugars, they are present in those cavities, they digest them. They produce a lot of acid, lactic acid, and that's why sometimes you taste sour. It's because the bacteria acting on the sugars that you've eaten, and it generates harmful things, toxic things, and creates a lot of plaque deposition inside our teeth, and that's what leads to tooth decay. Now, reproduction and nutrition. Prokaryotes have a very, very simple type of reproduction. It's called binary fission. It's like mitosis, but simpler than mitosis. In mitosis, there is a spindle fiber formation. There's complicated, the spindle fibers pull the chromatids, so the sister chromatids separate from each chromosome. In binary fission, there's nothing like that. So it's not very accurate. So a bacterium splitting like this, in mitosis, it will always be equal distribution of chromosomes, right? The chromosome number will double, and then the two daughter cells will get the exact same number of chromosomes because the chromosome number has doubled. However, in binary fission, sometimes the two daughter cells might end up getting unequal number of chromosomes because it's not perfect. There is no microtubule or spindle fiber formation. So some prokaryotes can actually survive unfavorable conditions by forming very thick covering around them. Those are called endospores. Now, because prokaryotes have existed on this earth for longer time than us, their survival mechanisms, pathways that transform energy and matter are more diverse. They have more survival mechanisms than us. Some of them, if required, they could harvest energy from, they don't even have to depend upon sunlight. They could harvest energy from ammonia, hydrogen sulfide. So the metabolic talents of prokaryotes make them excellent symbiotic partners, and a lot of the plants actually use, or a lot of even the animal, for example, this particular animal, this is what do you call it? I forgot, tube worm, right? Tube worm, they live in deep depths of sea. They don't get sunlight, so they have to derive energy, And what they do is they have a symbiotic relationship with bacteria. So these are sulfur bacteria. They harbor them, they house them inside their bodies, and the animals will absorb sulfur compounds from the water. The bacteria will help them to digest that, will convert them, okay, to convert carbon, will use these as an energy source, okay, because there's no light there, to convert carbon dioxide that they absorb from the seawater into organic molecules. What we do here, where there is light, is plants do it for us. They use sunlight, that is their source of energy, and they convert carbon dioxide to make glucose, and then we eat the plants. That's our source of energy. But in the depths of ocean, these particular tube worms, they use the bacteria which help them get the energy from sulfur. Now prokaryotic nutrition, so many different versions, okay. They can do photosynthesis. In addition to photosynthesis, they are also capable of nitrogen fixation. Nitrogen fixation means converting atmospheric nitrogen into a more usable form by plants. Our atmosphere has 78% nitrogen. However, nitrogen in this form cannot be used by plants. It has to be converted into nitrites and nitrates. That is how plants use it. And bacteria help them do it. So you see, prokaryotes, they might be simpler, they might be smaller. However, their survival mechanisms, the things that they can do, their metabolic pathways are way more diverse than us. So a symbiosis with a particular bacteria called cyanobacteria gives plants, such as this particular water fern that has been shown here, azolla, an advantage in nitrogen poor environment. Why? Because the bacteria will help them to get the nitrogen that they need. Now, recycling of materials, the ecological impact of prokaryotes and chemical recycling. Life depends upon recycling. If there is no recycling, as I said, if the dead bodies did not decompose, there would be no space for us. So life, existence or continuation of life depends upon the recycling of chemical elements between the biological and physical components of the world. So prokaryotes do this work for us. They promote the breakdown of organic wastes and dead organisms. And now that we've learned about this, human beings have started using the prokaryotes to do this for us. Okay? This process is known as bioremediation. This is the use of microorganisms to remove pollutants from water, air, or soil. So we use decomposers to treat our sewage, right? So all the sewage that goes in the treatment plants, there are a lot of bacteria that are doing the detox work for us. So they take care of it, they break down the big harmful chemicals or molecules into smaller harmless things, and then that can be recycled. Bioremediation took a leap forward. Another step up is when bacteria and other microorganisms were used to clean up toxic chemical spills in the ocean. So we do the mess up, we create spills, okay, ship sinks or something happens and there's a lot of oil and chemical spill in the water, and then we bring about the bacteria and prokaryotes to do the cleanup for us, and they are the ones helping us. Now we move to the categories of prokaryotes. So prokaryotes are broadly divided into these two groups, bacteria and archaea. And life in general is divided into bacteria, archaea, and eukarya. Let me show you first how we classify life, and then we will come back to this. Okay, so this is a major classification. All living organisms can be broadly grouped into prokaryotes and eukaryotes, right? So prokaryotes are archaea, bacteria, and what else? You will see. In the eukaryotes, these three groups you're familiar with these: the fungi, the plants, and the animals. Okay, all of these are multicellular. They can be unicellular as well. However, this particular group called protists, which is a catch-all category. So whatever eukaryotes they couldn't classify as fungus, they couldn't classify as plants, they couldn't classify as animals. Scientists, what they did was this group was created, and they put all of the eukaryotes, which are not plants, which are not animals, which are not fungi, they put it all in here, protists. And the protists, together with the prokaryotes, that means the archaea and the bacteria, all three, these three are together known as microbes. That means microbes can include bacteria, microbes can include archaea, and microbes will include protists. Although protists are not prokaryotes, protists are eukaryotes. That means they have membrane-bound organelles. They have a well-defined nucleus. So, what is the protist made up of? The protist is made up of what we call fungus-like protists, plant-like protists, and animal-like protists. Right? As I said, it's a catch-all category. It has everything. Miscellaneous. So, examples are, what is an example of a fungus-like protist is slime molds. All of us know slime molds. What is an example of a plant-like protist? For example, seaweed. Have you seen the seaweeds that they sell in Costco? Very good for, very good nutrition and expensive. And these are algae. These are algae. These are plant-like protists, and animal-like protists are called protozoans. And we will now learn about each of these categories. But this is the major classification. So, where were we? Okay, we were here. So now we know that life is categorized into bacteria, archaea, which are prokaryotes, and then this is the eukarya. And from the eukarya, we will be learning about the protists. So what are archaea? Archaea have a very wide range of habitats. One group of archaea can grow and can live in extremely high temperatures. They live in very hot water. They are called thermophiles. Thermophiles means heat lovers, okay? Another group is the extreme halophiles, means they are salt lovers. And both of these you can see if you go to Yellowstone National Park, you will see these, water puddles there that are bubbling. They're so hot, it's like boiling water. And you see different colors, greenish, yellowish. That's because of different bacterial colonies. And they just love that high heat. Okay, they say, there are bonding. Please do not go near them. If you just even dip your finger, all flesh will be gone, and when the bone gets dissolved. It's sometimes the salinity is so high, the temperature is so high, but the bacteria they can just survive in there. A third group of archaea, they are the methanogens, which live in oxygen devoid of environment which is no oxygen, and they give off methane as a waste product. So that's why methanogens, because they're giving off methanes. And they are usually abundant at the bottom of the lakes and also in the digestive tract of animals. For example, you might have seen these pipes, in landfills. These are for collecting gas generated by the methanogenic prokaryotes or archaea from a landfill. What will we do with this methane gas? Source of energy. To different forms of energy, just like your windmills, just like your solar panels, anything. Methane can be used to generate energy. So then we're done with archaea. That's one kind of microbe, right? That's a prokaryote. The next kind of prokaryote is bacteria. Okay, and bacteria that cause disease, they are commonly called pathogens. So most bacteria, most pathogenic bacteria, they produce a poison. That's how they give us disease. So what is that poison? Those are toxins. There are two types of toxins, exo and endo. So these two kinds of poisons are what give us diseases. Exo means outside. So these are proteins